many organisms pass their genes to their offspring through sexual reproduction.
This begins when two gametes unite to form an embryo that's genetically unique from the parent organisms.
The embryo then grows into an adult who in turn passes their genetic information onto their own offspring.
Gametes are formed through a process called meiosis.
In diploid organisms, cells have two copies of each chromosome. They undergo meiosis to produce haploid gametes, which only have one copy of each chromosome. These haploid gametes fuse to form a diploid embryo that grows into the adult cell.
The first division, Meiosis 1, results in two unique daughter cells that have half the amount of DNA as the parent cells.
PROPHASE 1.
In this stage, the DNA condenses to form chromosomes. Here, we see the duplicated sister chromatids joined together at the centromeres. Next, synapsis happens. Then, they exchange chromosomal material called crossing over or recombination. After crossing over, the sister chromatids for each chromosome are no longer identical to each other. This is the reason why no siblings aside from twins are genetically identical to each other. Then they attach to microtubules.
METAPHASE 1.
Here, they're aligned at the equator of the cell. Different alignments result in different combinations. Each time, meiosis occurs.
ANAPHASE 1.
During this phase, homologous chromosomes separate and migrate to two poles of the cell. It is important that the sister chromatids remain attached at their centromeres.
TELOPHASE 1.
The final steps are telophase 1 and cytokinesis. Here, the cell divides into two daughter cells. Each of these two cells now undergo meiosis 2.
The second division, Meiosis 2, results in four unique haploid cells that have only one copy of each chromosome.
PROPHASE 2. METAPHASE 2.
METAPHASE 2. They will align again at the center, the same as METAPHASE 1, also random.
ANAPHASE 2.
ANAPHASE 2. The sister chromatids are pulled apart while the cell elongated.
TELOPHASE 2.
In telophase 2, the nuclear membrane reforms and then the cytoplasm is divided into the two haploid daughter cells. The division is called cytokinesis.
Since meiosis 2 began with two cells and each of those cells were split into two cells, we now have four unique haploid cells at the end of meiosis. These cells are called gametes.